Good afternoon. My name is Matthias Reinhardt.
My name is Lily Jensen.
My name is Darshan.
And my name is Ethan Tran. Our group presentation is on what's essential for making essential oils. Before we get into the study, we want to define a couple of words for anyone who's unfamiliar. The DNA that we're going to be referring to is the genetic information that makes up the genes that code for proteins. And in this study, it's going to be CSLMI1 in the MYC5 gene. In a particular sequence of genes, there's a GCC box 6 regulatory element. This is just basically a binding section where an activator will bind to initiate the expression of that gene. The activator discussed in this study is CSDRNL. And then genetic mapping is just a term for a technique used that when you cross like parent generations and you analyze a descendant gene inheritance.
Citrus plants like lemon and oranges have patches in their skin that hold oils. These pockets called oil glands, benefit the plant in many ways. It includes protecting the plant against living things that eat plants and things that can cause sickness in the plant. These benefits are not limited to the plant. These oils can make flavoring and scents. They can serve as protector for other plants on farms. So this research paper we are presenting partly answers the question, what is the genetic recipe these citrus plants use to make these pockets that produce these oils? The researchers believe that a certain model and system can be used to understand this recipe. Imagine DNA as a long, twisted ladder with rungs made of different chemicals. If scientists wanted to add or change some of the twisted ladder, they can use CRISPR-Cas9. They can use the CRISPR-Cas9 method. Changing parts of the twisted ladder can help scientists determine the function of a specific part of a large ladder of DNA, such as CSLMI1. In this paper, scientists used CRISPR-Cas9 to cut out CSLMI1 to see its effects on oil glands. Another time they used this method was to determine if a certain part of DNA, the GCC box, decides how a plant develops oil glands. They cut out this specific part of the DNA and examined changes in the plant. And you can see these changes here. By finding out whether or not there was a pattern in the way CSLMI1 was present between the tip of the thorn versus the base on the thorn of the plant, they could figure out whether the specific DNA part played a role in the making of oil glands. To find out how much of the processed DNA part was present, they used a method called QRT-PCR. They used this multiple times throughout the paper. Along with QRT-PCR, after collecting pieces of the base and the tip of the thorn, they used a method that allows them to see where a particular message, CSLMI1, was present within these pieces of thorn. This method is called RNA in situ hybridization, and it is like a camera that takes pictures of where this message is and even smaller parts of the pieces of the thorn that was studied. By doing this, they can see if there were any differences between where this message was located in small pieces of the base and tip that they studied. If there was a difference, it lets them understand how the plant uses the message differently in different parts of the thorn. This experiment was also used in a similar way throughout the paper.
As for the results of the experiment, Figure 1 compares two different types of kumquat, here called HP and RA. As seen in images A and B, HP has no little circles in its leaves, meaning that it has no oil glands and very few on the whole fruit. RA, however, does express oil glands. The leaves in image C show the importance of oil glands as a defense mechanism. It can be seen that the RA leaves are still whole after 12 hours, while the HP leaves have been eaten by worms. The difference between the phenotypes of these two fruits is what lets the researchers figure out which genes are related to oil gland production. As seen on the CRISPR slide, genes can be removed from a strand of DNA. Part E of this image is showing both the genotypes and phenotypes of a wild type, or unedited, leaf, and 3 leaves with different percentages of the CSLMI gene removed. F and G are comparing the number of glands as well as how much LMI1 is being expressed. We can look at the heights of the bars and see that the leaf with the completely removed gene has the largest number of glands. A regulatory pathway is something like a domino effect, and it is what the paper was focused on discovering in the end. It's like a domino effect that happens inside of cells. In a pathway like this one, each enzyme and protein and gene needs to do its job before the next step in the process can happen. The data from this paper in particular indicates that the process of oil gland development starts in the outer layer of a fruit with DRNL acting as a flag or a flare so that CSLMI1 knows to start doing its job. Because CSLMI1 was activated in the first step, it can now help create oil glands in the skin, and this causes MYC5 to be expressed. MYC5 then causes the oil glands to be fully expressed, and it begins the essential oil production process, as can be seen in this timeline of cell growth.
As we have established, CSLMI1 it plays an imperative role in the appearance of the leaves and the existence of oil glands in citrus plants. This has been identified as a transcription factor, which is pretty much a protein that controls which genes are expressed during DNA replication. CSLMI1 has been identified to play a role in making the leaves serrated and having glands to secrete oils. This study has also identified other elements that impact the citrus species having oil glands as well. Another element that has been identified is the GCC box, which is identified as a promoter to activate CSLMI1 and starts the entire process. The experimenters used the CRISPR-Cas9 gene editing tool to determine that the GCC box and CSLMI1 are necessary for the serrated leaves and oil glands to be produced in these citrus plants. These findings are important because they deepen our current understanding of how essential oils are created and secreted in citrus plants. These findings can lead to the development of protection for these citrus plants in the coming years.
At the end of the study, the researchers concluded that the formation of oil gland development is initiated by the binding of transcriptional activator CSDRNL to the GCC box cis regulatory element, which induces the transcription of HDZIP1 family transcription factor CSLMI1. This CSLMI1 activates CSMYC5, whose job is to promote the production of oil gland cells and controls the expression of genes that participate in the natural creation of essential oils. This entire molecular pathway is not only operational in leaves, but also in fruits. The discovery of this formation process of such secretory cavities and accumulation of secondary metabolites is important because it is necessary for protecting plants against herbivores. It can also be manipulated for producing essential oils involved in scent and flavoring. To build upon the findings of the study, further research could investigate how this specific pathway, as well as similar pathways in other leaves and fruits, can be used to synthesize new essential oils from sources that perhaps don't naturally produce their own. The researchers indicate that perhaps the artificial implementation of such pathways in some leaves and fruits could lead to the protection of those plants and even the essential oil scent or flavor of a plant that doesn't normally have one. And this is our source, our article that's being used. And that is it.
Thank you.